Hi everyone, this is your instructor, Christina Howard, for Biology 242. That is the second half of Human Anatomy and Physiology at Clark College. So where we left off, we were talking about how hormones act on the cells that are their target cells. So let's first switch over to Blackboard and define target cells. So, oops, and I gotta do this first. There we go. A target cell for a hormone is any cell expressing receptors for that hormone. And that's an important concept because you can change the effect of a hormone in two ways. One, you can change the amount of hormones circulating. And two, if you don't want to change the amount of hormones circulating, a change in sensitivity can be achieved by a cell increasing or decreasing the number of receptors. So I'm going to draw a cell. Actually, let's draw two cells. So let's give our cells a nucleus. So you've got a red cell and a purple one. So the purple cell has receptors for one hormone. And these little Y-shaped guys are receptors. And the red cell has receptors for a different hormone. So if I have a gland somewhere off over here, I'm just going to make a blobby gland, G. So let's say this gland secretes purple hormones. And those are going to travel in the bloodstream over to these target cells. The only one of these two cells that is capable of responding to the signal that that hormone is indicating is the one with the receptors for it. So even though this cell is surrounded on all sides by this hormone, it will never respond to the hormone because it doesn't have the appropriate receptors for it. It has receptors for a different hormone, which is why it won't respond to the purple one. So receptors really, really matter, and it's important to keep receptors in mind when you're considering the actions of hormones because they are just as important as the hormones themselves. Okay, so let's get back to this, and I'm going to go ahead and erase my little thing here. So what do they do? This is a list. It's not exhaustive, meaning it's incomplete, and it's also very, very general, and that's on purpose. The reason for that is endocrinology really could be a class all of its own. Hormones and their actions are incredibly complicated, and they're part of very complex sets of feedback loops, and we just don't have the time to go into every single one of those. I wish we did because it's fascinating, but we don't. So for some of the next portion of the lecture, you might feel at times like I'm being a little bit vague, but that's on purpose. I'm glossing over some stuff because they're just details that we don't have time for in the class. Feel free to ask me about them either on Slack or by email or what have you. Just be aware, hey, it's very possible that we're not going to get to them as a testable item in 242. Okay, so what do they do in general? Well, they change enzyme activity. And that can mean increasing it or decreasing it. It could be either. They can also open or close ion channels. So one significance of that particular bullet point might mean, let's say they close ion channels. If they close ion channels, like let's maybe say voltage-gated calcium channels, that might inhibit a cell from releasing neurotransmitter, for example. So that's one example of how that can work. They can also activate genes. So remember from Biology 160, genes code for proteins. Many proteins are enzymes. And so they can either cause the increased synthesis or decreased synthesis of enzymes and structural proteins, i.e. collagen, keratin, etc. So a great example of genes and changing synthesis of structural proteins that happen in response to hormones is the hormone dihydrotestosterone stimulates facial hair growth, during the onset of puberty in XY individuals that have a lot of circulating androgens all of a sudden. So that's one example. They can also increase or decrease the synthesis rate. So they may not fully turn on or off protein synthesis, but they can speed it up or slow it down by stimulating or suppressing gene transcription and mRNA translation. So they can act at any level of the central dogma of molecular biology by just sort of tweaking the speed of things. Okay, so that was a little bit of a review from last time. That's kind of where we ended. So now we need to talk about hormone mechanisms of action, meaning how do they do their work? And again, we're going to be kind of general here because, which protein receptor it is. There are many subtypes of protein receptors. Remember from our discussions of the autonomic nervous system how there were lots and lots of different norepinephrine receptors. For each hormone, there's more than one receptor kind as well. For example, melatonin has 3 known receptor types in the brain alone. So that's just an example. The mechanism of action is going to, in part, be contingent upon, let's underline that here, depends on hormone class. And I here mean chemical class, because the solubility of the hormone in fat or in water is going to both determine how it's carried in the blood, as we discussed previously, but also, also determine, once the hormone gets to the cell, whether it binds to a receptor on the plasma membranes on the edge, or whether it binds to a receptor that's inside of the cell. So the hormone class is going to determine whether the hormone can get through the cell membrane or not. So ones that are water soluble tend to bind to a receptor on the outside of the cell, and that receptor is going to be positioned in the plasma membrane. This activates an intracellular second messenger like cyclic AMP. So remember adenylate cyclase. Let's write that down. Adenylate cyclase is the enzyme that takes ATP and turns it into cyclic AMP, right? So that's what that does. And the other kind of second messenger is calcium ions, and those do a variety of things, including, change permeability of membranes. They can function as a signaling molecule if they're complexed with calmodulin, all kinds of stuff. If the hormone in question is fat soluble, it will cruise right through the membrane. It'll skip the transmembrane proteins because it doesn't need to go there, and it will do something on the inside of the cell. So these typically bind to receptors in the cytoplasm or in the nucleus of the cell, and that changes some intracellular activity. Again, vague on purpose because it really depends on the cell. Oh, I was talking while writing and failed to spell depends right. Depends, there we go, on the cell. So which cell? But also among cells, which receptors are present. So it depends on a couple things. So again, I'm being vague on purpose because what that really means is going to change a lot from situation to situation. Okay, so let's go to, so mechanism of action. Remember for the catecholamines, so even though catecholamines are cholesterol derived, they do tend to stick to cell membrane receptors. And this should be no surprise because we talked about alpha and beta adrenergic receptors last term in 241. So if you need a review of those, go find your 241 autonomic nervous system slides, and there's plenty of stuff in there for you. Peptide hormones, unsurprisingly, because of course those are water soluble. And interestingly, eicosanoids. So although those are technically lipid derived as well, they're also an exception. They don't bind to inside of cell stuff, they bind to outside of cell stuff. So for these guys, all known receptors are in the cell membrane. And binding of a hormone to the receptor, so the protein receptor and hormone complex, activates the G protein. And then inside of the cell, the G protein changes the concentration of cyclic AMP. So sometimes it makes it rise, sometimes it makes it fall. And then also can change the concentration of calcium ions. And I'm going to put square brackets around these because square brackets is how we denote concentration in biology. Okay, so mechanisms of hormone action. These are very important, and I do expect you to understand these. So, effects on cyclic AMP or effects on calcium. Those are our two major classes. Within effects on cyclic AMP, we've got G proteins activating adenylate cyclase, which means they're going to, this one, increase CAMP. And that's going to act as a second messenger, and it's going to activate enzymes or open ion channels. And I want to take a moment here to define kinase. So cyclic AMP uses kinases to activate enzymes. So let's go to Blackboard here for a minute. Okay, so let's talk about kinases. I brought up the Blackboard here, and I'm just going to draw out some basics about kinase, and we're going to start with a little bit of terminology. So what I'm going to do here is draw while I'm talking, and hopefully that will help clear up kinases and their opposite counterpart, which are phosphatases. So let's look at these. So we're talking about this stuff in the context of the actions of hormones, but really kinases and phosphatases are active parts of regulating nearly all cell processes. So this is just an important thing to know about cells in general. So kinases, let's look at this word here first. ACE means it's an enzyme, and all enzymes are proteins. So this is a chain of amino acids that's folded up in a particular way to catalyze a specific reaction. So following that, enzyme is a protein that catalyzes a reaction, which I'm going to abbreviate RXN. So there's a general form for kinase, which is, general equation for kinases, which is applicable to all kinases. So I'm going to draw that chemical equation out. Just in a very general sense, you have a broad idea of what kinases do. I want you to keep in mind while I'm talking that this is not one particular kinase. This is any enzyme that is designated a kinase, which is a larger group of enzymes. So let's also define kinase specifically. So I said it's an enzyme, and specifically it's an enzyme that, here's that word again, catalyzes transfer of a phosphate group. From a high energy donating phosphate molecule, usually ATP, because the bonds between the phosphates in adenosine triphosphate have high energy. And then where do they transfer it to? Well, whatever the specific substrate in the context of this particular kinase is. So I'm going to say phosphate accepting and reaction specific molecules. So if you go back and look at the signaling cascades figure in your textbook that shows you various second messenger cascades, both amplification and suppression, you're going to see that kinases are involved. So the generalized form for a kinase reaction is, let's say we have kinase plus ATP, and I'm going to use a symbol here instead of a word. I'm going to say pink blob. Let me color in my pink blob. So in my pink blob, the active site is blocked. So it's right there. Yellow is the active site. So here I have a protein. Maybe it's an enzyme. I'm saying it has an active site, so it probably is. But currently this protein is closed and can't act. So with kinase, we get the reaction. And remember, enzymes catalyze reactions but are unchanged by the reaction. So it's not usual to put them in the equation in this sense, but I'm doing it so that you can see that both sides contain the same kinase and that kinase doesn't undergo any chemical changes as a result of the reaction. So now we have a DP because we've stolen a phosphate, or sometimes we even have cyclic AMP, which is mono, and we have an opened or now active protein, maybe with one or two phosphate groups attached to it. Sometimes not, though. So in your situation, we've got cyclic AMP, which feeds into the amplification process. So a hormone binds on the outside of a cell. That hormone never enters the cell, but instead kicks off a series of chain reactions that activate kinases and activate cyclic AMP, and those series of events produce a signaling cascade, a cascade being one enzyme activates another, and those enzymes set other processes in motion, and that is how the signal is amplified inside of the cell as a result of the hormone binding on the outside of the cell. So kinases are used to activate enzymes, and also cyclic AMP can open ion channels as well. So the net result is enzymes activated or ion channels are opened, and in this case we have acceleration of the cell's metabolism. So let's discuss that now. So here we have amplification. So low concentrations, meaning the number of parts per million or even parts per billion in the blood of water-soluble hormones, even very small concentrations of those are capable of very large effect sizes because the signal is amplified when it gets to the cell. And that's because one activated G protein can generate thousands and thousands and thousands of cyclic AMP. So you suddenly have cyclic AMP levels in the cell. So let's draw a chart here. So this is amount of cyclic AMP. This is time. And I'm going to make this point right here be when the G protein is activated. So maybe, let me change colors. So maybe a hormone level is, or a cyclic AMP level inside the cell is relatively low. Then a hormone binds, the G protein is activated, and the cyclic AMP amount rapidly rises after that. So that is this. And we know that cyclic AMP activates protein kinases. Protein kinases activate enzymes, and enzymes are then able to catalyze chemical reactions. So adenylate cyclase, again, like I said, produces thousands of second messengers, and we're only looking at one protein receptor and one G protein here, so one adenylate cyclase. There can be, remember, many, many thousands, tens of thousands, 200,000 even, receptors for a hormone on a particular cell. So amplify that times the number of receptors, and you can see that the amount of cyclic AMP in the cell is going to raise very, very quickly. Okay. Other cyclic AMP effects, it can inhibit as well. So in the other case, you can reduce enzyme activity. So for example, over here we have G protein activation converting cyclic AMP back into regular AMP. So that's going to reduce enzyme activity. Oops. And I want to stress, so reducing enzyme activity is important for certain cells. So maybe you want to turn off part of a feedback loop for a little bit, or maybe you need to make a cell less sensitive to something. This is going to accomplish that goal. So this decrease has an inhibitory effect on a cell. One piece of the puzzle that's missing is PDE. So kind of the opposite of adenylate cyclase. This stands for this word. Phosphodiesterase. So this is an enzyme that basically turns cyclic AMP into a non-signaling form, which is just regular adenosine monophosphate. Okay, so phospholipase C. This is the one that activates calcium. So this is a different situation. We're not using cyclic AMP anymore. Instead, same process happens. So a protein binds a hormone receptor, or excuse me, a hormone binds a protein receptor that activates our G protein. And then we've got phospholipase C, which is what PLC stands for. DAG stands for diglycerol, and IP3. I'm actually not going to make you worry about IP3. So PLC, phospholipase C, is the biggest one that we're going to worry about. So that's the one you should focus your studies on. These other two, why don't you just go ahead and ignore them. So one thing this does is open calcium channels, and that lets more calcium in. The other thing is it can take calcium that has been stored somewhere in the cell and release it into the cytoplasm. In both cases, calcium is going to complex with calmodulin, and the complex of calcium plus calmodulin activates enzymes. So think of calcium as the on switch for calmodulin, and then calmodulin is capable of traveling around and turning on enzymes wherever it goes. Okay, steroid hormones in general, and these tend to be sex steroid hormones, or in some cases stress hormones, so corticosterone, cortisol, etc. These typically bind to receptors either in the cytoplasm or DNA-bound ones. We're using the cytoplasmic ones right now, but again, remember, I have to be vague about this stuff because we don't have time to go into the specifics of every single thing. So these activate genes, they stimulate protein synthesis, and the result is that they're going to change cell activity and cell structure, and in both of these cases, via changing gene expression. So the turning of DNA into RNA and then turning RNA into proteins, they're going to change how that happens in a cell. And that is what's going to inform the target cell's response. But again, being intentionally vague here because what exactly that looks like depends on which proteins are being synthesized and what those proteins change inside of the cell. So again, context dependent. Thyroid hormones are a little weird. So even though they are amine hormones, so amino acid hormones, monoamine means one amino acid. So the monoamine hormones are hormones that are derived, again, from one amino acid, so either tyrosine or tryptophan. So these guys increase ATP production in general, or they activate genes altering cell activity. So they can do both. They are transported across the cell membrane by some specific processes. I'm not going to go into the details of those, just know that they get help crossing the membrane. And then, unsurprisingly, since they're increasing ATP production, they're going to bind to receptors on mitochondria. And the mitochondria are going to respond by pumping out a lot more ATP. They can also do an action that is very similar to that of steroid hormones, which is to bind a receptor inside of the nucleus, and that complex sits on DNA and promotes gene activation and transcription. So changes in energy budget as a result of thyroid hormone are this domain, and other things that thyroid hormone does. For example, thyroid hormone levels are critical for development of a healthy fetal nervous system. That particular process is more driven by this, controlling the synthesis of proteins. Okay, so let's summarize. Hormone effects on target cells. They're going to change enzyme activity in a variety of ways. They can up or down regulate it. They can open or close ion channels. They can change gene expression. They can do all of these things. It really just depends on which hormone and which receptor. Okay, so this activity was an in-class activity, but instead it's going to be part of your case study activity. So stay tuned for that. I'll also give you some little demonstrations here and there in some other videos about endocrine feedback loops and the like. So we'll talk more about that later. So before we move on, I will say humoral factors means, let me get my pen here. Humoral here means in the plasma. So for example, this is one that you're familiar with from our discussion of the skeletal system. You know that if blood calcium, plasma calcium drops, there is going to be an increase in the secretion of parathyroid hormone. A lot of times, hormones cause other hormones to be secreted. This is very common. So, for example, the releasing hormones of the hypothalamus cause the anterior pituitary to release hormones as well. And neural impulses can also do this. So, for example, the release of oxytocin, which is a posterior pituitary hormone, during labor and childbirth or during sexual activity is actually stimulated by neural impulses. So one thing to keep in mind, and one thing that's important to understand, is that hormones are secreted via negative feedback mechanisms. So what that means is a given hormone will circulate back to whatever caused its production in the first place and inhibit its own production. And this is an important concept because what this allows is for hormonal responses. Let me grab a pen here. So when you have negative feedback, here's what a typical hormone release event looks like. I'm gonna get green. Okay, so actually let me get white. Label my axes. Always label your axes. Don't be like me. Okay, so this is amount of hormone. This is time. And here we have our curve. So let's say something causes the release of this hormone. So after the releasing incident. So maybe it's pretty low levels in the plasma, and then it sort of begins to climb. And it hits a peak, but remember, this hormone is circulating in the blood, so it's also going to be circulating back to whatever caused its release. And that thing, usually it's a gland, sometimes it's not, is going to express receptors for that hormone and stop sending out the signal for the release of that hormone, which is going to cause the circulating hormone levels to fall back down. And unless there's an additional impulse for hormone secretion, what we get is a pulse. So a little infusion of hormone into the bloodstream for a short amount of time to do a specific task, and then it's turned off again because it's no longer needed. So the significance of negative feedback is really about being able to release hormones on demand and turn off their release when they're no longer needed. So it's just as important to be able to turn off a signal as it is to be able to turn on a signal. Okay. Oopsie. There we go. All right, so for instance, we're going to talk about how the hypothalamus controls endocrine function. So the hypothalamus is part of the diencephalon, as you guys remember from the nervous system. And it has many, many regulatory nuclei. We're not going to talk about those so much, but rather we're going to discuss how the hypothalamus controls the anterior and posterior pituitary, and how it controls the secretion of epinephrine and norepinephrine. But before I do that, I'm going to run over and close my window because there is a very loud sound happening outside. I'm not quite sure what it is. So hang on for just a second. There, that's better. Sounds like somebody's dragging a metal sheet over the asphalt. It's a very obnoxious sound. Hopefully it's not making it into my mic very much. All right. So, there are two ways that the hypothalamus controls the pituitary. One is by releasing or inhibiting hormones. So one is by humoral factors. The other is via neural impulses. So which of those is occurring depends on whether you're talking about release of antidiuretic hormone and oxytocin, from the posterior pituitary, this is what I meant to do, or whether you're talking about release of what are called the tropic hormones from the anterior pituitary, or catecholamines from the adrenal medulla. So we'll get more into that in a little bit. I do want to stress, though, that more complicated patterns of control, so more complicated than negative feedback, are not uncommon. So one of those, and these are all things that are determined experimentally, I want to remind you. So these facts that I'm presenting come from lots of experimental data. So one finding is that for some situations, the same hormone can have a different effect depending on its concentration. So it can do one thing at a low concentration and a different thing at a high concentration. I already showed you the burst pattern. So a lot of times a hormone isn't secreted constantly at a particular level, but rather you get release of a hormone into the bloodstream so that the serum concentration of hormone rises and then it falls back down. That's a pulse. So sometimes a hormone is released every 3 hours. Sometimes they are released every 30 minutes. It really just depends on the context. Okay, so anatomy of the hypophysis, which is another way to say the pituitary gland. So let's write that down. AKA pituitary. So to do this, I'm going to tell you a little story. And that is how the hypothalamus and pituitary talk to each other, but more importantly, how they came to be. So to go over, let's go to Blackboard. So this is now when I need to draw a little embryo. So during embryonic development, so there's the little eye bud, we have a big old head, and then a little mouth, and a little leggy, and a little arm bud. And they typically have a really big belly. So here's my embryo. It's really small. So remember from our discussions of the nervous system, and I'm going to pick a different color for this. Let's do orange. The brain is still sort of developing. So we've got the telencephalon differentiating, and I'm not going to make the telencephalon and diencephalon and rhombencephalon and so forth all different. But rather, I'm just going to make the CNS be sort of a primitive shape. So, during fetal development, a stalk begins to grow downward from the hypothalamus. Also, during fetal development, you get the development of an embryonic pharynx. So, your pharynx is the muscular tube or funnel that channels air through your nose down into your lungs and food down through your mouth into your esophagus. So the embryonic pharynx, specifically at the roof of the mouth, which I'm drawing here. So here we have our little pharynx, and then maybe a little tummy and some early intestines. The embryonic pharynx starts to pouch out at the back of the roof of the mouth. And this structure is called Rathke's pouch. So Rathke's pouch starts to reach up. The infundibulum of the hypothalamus starts to reach down. So we've got neural tissue reaching down towards epithelial tissue, which is reaching up. Again, once ossification gets underway, these two structures end up being enclosed in the sphenoid bone, such that from this process, down growth of the hypothalamus and upgrowth of Rathke's pouch from the embryonic pharynx, you get the following. So let me draw a sella turcica. And then you get this, where you've got an anterior pituitary. So this is leftovers of Rathke's pouch. It's epithelial in nature, so it looks like a glandular epithelium. And then growing down from the hypothalamus, we have the posterior pituitary, and this is nervous tissue because it came from the diencephalon. And then the bulbous part of the end of the anterior pituitary, the part that came from Rathke's pouch, the bulbous part is called the pars distalis. And then the part that's wrapping around the stalk is called the pars tuberalis. And then down here, for the bulbous end of the posterior pituitary, we have pars nervosa. And then the stalk is the infundibulum. So that is the embryonic origin of the pituitary gland. And what you're going to notice in histology in lab is that the appearance of the tissues in the pituitary look very different. So the posterior pituitary, the pars nervosa of it, is very different in histological appearance and cytoarchitecture from the pars distalis. All right, goodbye embryo. Let's go back to presentation as usual. Okay. So we went through that. So the first thing we're going to talk about is how the hypothalamus talks to the anterior pituitary, because that is very different from how it talks to the posterior. So what you're going to notice here are blood vessels. So we've got two arteries, the superior and inferior hypophyseal arteries. The superior one gives rise to a capillary bed, which is here. So that's the primary capillary bed. Down here, we have a secondary capillary bed connected to the first one by what are called portal veins. So here's what the hypothalamus does. The supraoptic and paraventricular nuclei are responsible for release of hormones down here. So we're not going to worry about them yet. Neurosecretory axons from here, these, notice they're not synapsing on other neurons. They're synapsing on the bloodstream. So these are going to secrete releasing hormones or inhibiting hormones into this first capillary network. Those releasing or inhibiting hormones are going to travel a very short distance to this capillary network where they will diffuse out and act on the cells of the anterior pituitary. Those hormones will then cause the release or inhibition of release of hormones from the anterior pituitary itself. So this is how a hormone can cause the release of another hormone. So a portal system is just two capillary beds in sequence, and it gets a special name because in the body these are pretty rare. So you don't have them, usually it just goes artery, capillary bed, vein, and then back to the heart. Usually there's not two capillary beds in a row. This is a notable exception. So I'll show you a feedback loop that involves these in a little bit, but not quite yet. So before we do that, I'm going to actually, let's do this first. I usually do this in a different order, but I actually think I'm going to change it up because I'm not teaching in person and I have the luxury of some different teaching methods when I teach remotely. So that actually clears up some space for me to do some different things. Yay! Okay, so I talked about releasing and inhibiting hormones. And these are stored and released like neurotransmitters. So what that means is by action potentials and then via exocytosis. So as you can see here, we've got the hypothalamus producing a releasing hormone. That releasing hormone is released into the primary capillary bed and travels down via the portal veins to the adenohypophysis. The adenohypophysis, in response to the releasing hormone, is going to release hormone number 1. So this hormone right here is not the same as the releasing hormone. The releasing hormone caused the release of this hormone. That hormone is going to travel to an endocrine gland and act on hormone number 2. Hormone number 2 is going to travel in the blood to its target cells, but also it's going to travel in the blood back to the adenohypophysis and back to the hypothalamus to inhibit its own production by inhibiting the production of the releasing hormone and inhibiting the production of hormone number 1. So let's look at a specific example. Let me just make sure I don't have a slide for it first. I prefer to draw it out. Okay. So I will do this. We have another example coming up because, again, repetition is our friend when we're just learning this stuff. And as you can see, the feedback loops are complicated. So let me just draw my little diagram here first. So what I'm going to do is make myself a hypothalamus. And then I'm going to ignore the posterior pituitary for now and just focus on the anterior pituitary. That's going to be the one in pink. So let's say hypothalamus releases CRH, which is corticotropin releasing hormone. Corticotropin releasing hormone acts on the anterior pituitary. In response to that, the anterior pituitary is going to release ACTH, adrenocorticotropic hormone. And I'm not going to write that one out. You have it in your book, so you can go find out how it's spelled. It's just a long one. So what I'm going to do now is I'm going to go make these arrows into green. So now I need to draw myself an adrenal gland. And adrenocorticotropic hormone means a hormone that acts on the adrenal gland's cortex. That's what cortico means. So adrenocorticotropic hormone is going to cause the release of certain other hormones. I'm going to pick one arbitrarily. So I'm going to say it causes the release of cortisol. Why not? So cortisol is a hormone that is considered to be a stress hormone, and specifically cortisol's responsibility is to change the way the body uses fuel. And that's because under stressful events, fuel use needs change, and so the way that the body deals with fuel and its budgeting of fuel has to also change. So cortisol is going to go do whatever it does to its target cells. So specifically, it likes to act on fat, for example. So let's draw ourselves some adipocytes here. Now I'm going to switch to red. So let's say a stressor was experienced and cortisol levels rose. Cortisol is going to circulate in the blood back to inhibit the release of ACTH. So actually, maybe I should draw this arrow this way. So a red line with a T end means inhibit. So this is going to prevent the release of ACTH from the anterior pituitary. It's also going to feed back to the hypothalamus and block the release of CRH. So what this would look like is you'd see a transient rise in cortisol, and then the level of cortisol would come back down unless there is an additional stressor to cause another pulse of CRH and ACTH release. So this is how hormone negative feedback loops work, especially those which involve the hypothalamus and anterior pituitary. Okay, so I just drew you one axis. This is an image that's diagramming the relationship of the pituitary hormones with their target tissues, and it also mentions the release hormones. So, for example, I just gave you corticotropin-releasing hormone causes ACTH release, which causes glucocorticoids, which are cortisol, the one I used, and corticosterone, which is the other one. So you can see there's multiple axes. So for example, the hypothalamic-pituitary adrenal axis, the relationship between those is called the HPA axis. The relationship between the hypothalamus, the pituitary, and the gonads is called the HPG axis, so hypothalamic-pituitary gonad axis. So what you need to start practicing, and I'll help you do this in Zoom sessions, and I'll help you do this using your activities, is to begin practicing with your hormones. So know which releasing hormones cause the release of which pituitary hormones, and know what those pituitary hormones do in general. So you need to be able to reproduce parts of this image here. Here we have another example. So, thyroid stimulating hormone. So, thyrotropin releasing hormone acts on the anterior pituitary. That causes the release of thyroid stimulating hormone, which does what it sounds like. It stimulates the thyroid. So, thyroid stimulating hormone is also called thyrotropin. And its effect is, of course, to stimulate the thyroid gland, like the name says. So the effect of this is you get a stimulation of thyroid hormone release, but then that thyroid hormone is going to feed back and inhibit the production of thyroid stimulating hormone and thyrotropin releasing hormone. So just like the negative feedback loop I just showed you. So this is a nice table, which I'm not going to read to you as usual, but rather I'm just using it to say if you want a nice summary when you're starting to sort of learn and organize your knowledge about the relationship between the hypothalamic releasing or inhibiting hormones and the hormone themselves and then the targets, this is a good place to start. It's organized really nicely. Okay, so speaking of the thyroid gland, thyrotropins cause the release of thyroid hormones. Thyroid hormones come in more than one variety. So there's two hormones that come from the follicular cells. So remember, if you already watched the lab video, you'll know what these look like. The follicles are hollow spheres of simple cuboidal epithelium, and the cells of that epithelium make thyroglobulins, as well as making thyroid hormone, and then put those into the follicle cavity. So the two kinds are triiodothyronine and tetraiodothyronine. So tri and tetra refer to how many iodines the molecule has in it. So either 3 iodines or 4. T3 is far more potent, so it has a very much stronger effect on target cells than T4 does. But it's easy to convert T4 into T3 just by removing an iodine. So T4 represents a pool of convertible T3. So you can convert T4 into T3 really easily. You just got to take an iodine off. So this is stored in follicles, as you saw, and those follicles look pink with an H&E stain. These absolutely require dietary iodine for production. So what we're going to look at next is the consequence of what happens when you break that feedback loop. Come on. I want to go to the next slide. Thank you. Okay, so this is a person's neck. And this large mass here is what is called an endemic goiter. So this is not fat and it's not a tumor. This is the thyroid gland. Specifically, we call it a hypertrophic. Hypertrophic thyroid gland. So endemic goiter is caused by inadequate, iodine. So if you've ever purchased salt from the grocery store and you've looked at it and you've like, well, I have a choice between iodized salt and non-iodized salt. Why would there be iodine in salt at all? Why would anybody put it in there? Well, the answer is foods containing iodine are all over the place, but not everybody eats them. So in countries, for example, where the primary source of caloric nutrition, so where you get your calories, is mostly like cereal grains, those don't have much iodine in them compared with dark leafy green vegetables, for example. So putting iodine in salt's delicious, is a good way to get people to just consume iodine with their salt. So what does this mean? Let's draw a axis again. So here we have our hypothalamus, and let's draw our anterior pituitary. Okay, so hypothalamus is going to cause the release of thyrotropin releasing hormone, and that is going to act on the anterior pituitary to cause the release of thyroid stimulating hormone. And that's going to act, let's draw ourselves a thyroid, on the thyroid gland. And the thyroid gland is, in response, going to release T3 and T4. Right? Wrong. So remember, and I'm going to use yellow for this because iodine is yellow, so it's yellowish orange. So need iodine. So what happens if the thyroid gland does not have enough iodine to make T3 or T4? Let me erase all these little dots. So now we have a thyroid gland that is unable to make T3 or T4. So it's going to say something along the lines of, I can't. And it has maybe a little frowny face because it can't do its job. So the hypothalamus is telling the thyroid, or telling the pituitary to make TSH. TSH is telling the thyroid to make thyroid hormone, but the poor thyroid does not have enough ingredients to make thyroid hormone, which means that ordinarily you would have negative feedback to inhibit the production of TRH and inhibit the production of TSH. Now you don't, because there's no thyroid hormone being produced. So what that makes the hypothalamus think is, it just thinks, I must not have gotten the message across. I better release even more TRH. And that causes the pituitary is not affected by iodine amounts, it can still make hormone. That causes the pituitary to make a lot more TSH. And increases in TSH are going to further stimulate the thyroid gland. But the thyroid gland is like, again, I can't make that stuff. I don't have the ingredients. I'm trying. So what we have is these two levels of TRH and levels of TSH, very high levels, because they are under-inhibited. And we have a frustrated thyroid that is trying very hard but can't make what it's supposed to do, so the thyroid is overstimulated. It's being stimulated by TSH too much. So, as the thyroid tries and tries and tries to make thyroid hormone, it's making the parts of the molecule that don't have iodine and storing them, but it's not adding the iodine, so it's not releasing anything. This causes the tissue to grow. And grow. Because it's trying and it's like, well, maybe if I add more tissue, I can solve the problem. That doesn't solve the problem, but it does give you a big, huge thyroid gland. So this is how a lack of a nutrient breaks an endocrine feedback loop and causes overstimulation and therefore an increase in size of a particular gland. So that's what causes a goiter. This is the little thyroid being like, can't make T3 and T4. Frowny face. So we're going to practice with feedback loops like that on the case study activities. So what does thyroid hormone do to cells? Well, basically it turns up the rate of ATP production. So that's how it works. So if you're making more ATP and you think back to what mitochondria do, remember their primary thing is oxidative phosphorylation. So they use oxygen as a final electron acceptor to pump protons into the intermembrane space of a mitochondrion, and that creates a proton gradient which powers ATP synthase. So that's how we do that. What that means is if you see high levels of thyroid hormone, you could expect increased levels of O2 consumption and increased levels of ATP production and ATP use. So the way we say that the short way is just say the net effect of thyroid hormone is that it increases metabolism. So it's going to increase protein synthesis, it's going to increase glycolysis, and it's also going to increase lipolysis. So the splitting of sugar and the breaking of triglycerides from adipose tissue in order to produce energy. It also increases your heart rate and force of contraction, because if you need more O2, the O2 delivery system has to keep up. And, unsurprisingly, thyroid hormone also regulates growth and development. And that doesn't really come as a surprise, right? Because when you're growing, you're doing a lot of mitosis. Guess what mitosis costs a lot of? ATP, right? So you would expect increased levels of thyroid hormone in a person who is actively building new tissue. So earlier I mentioned that thyroid hormone is critical for the proper development of the fetal nervous system. Thyroid hormone is also critical for growth and development during childhood. Interestingly, thyroid hormone also has what's called a permissive effect. So a permissive effect is when the binding of one hormone to a cell or tissue makes that cell or tissue more sensitive to a subsequent or different hormone. So thyroid hormone causes an increase in the production of beta receptors on cells, which causes an increase in sensitivity to sympathetic stimulation in those cells. Another thing it does is stimulates the kidney to release erythropoietin. Erythropoietin is a hormone released by the kidney, and that hormone basically causes increases in red blood cell production. Okay. Suprarenal or adrenal glands. So I will talk more about the histological differences between the zones in the lecture for lab, but for now let's talk about the suprarenal glands because we're about to start talking about the human stress response. So the outer cortex secretes corticoid hormones. Corticoid refers to their being from the cortex. And these include mineralocorticoids and glucocorticoids, which we'll discuss. The inner medulla secretes catecholamines. So remember, the medulla of the adrenal gland is where those weirdo annexonic neurons live, and they only secrete epinephrine and norepinephrine. Unsurprisingly, because these hormones are dumped directly into the blood, the adrenal glands are highly vascularized, so they have a really robust blood supply. So, let's talk about the mineralocorticoids. We're primarily going to talk about aldosterone. There are others, but worry about aldosterone only. So, the zona glomerulosa releases aldosterone. So, this is a hormone that does a bunch of things. Primarily, what it's going to do is change water via ions. So, what it does is it causes the kidney to hold back more sodium. So sodium that you would ordinarily be peeing out, you're now keeping. And remember, the golden rule of osmosis is that water follows salt. So if you're saving your sodium, water's going to follow it, meaning that you're also saving water. That water gets returned to your blood and increases your blood volume. However, this is at the expense of both protons and potassium. So in someone with chronic aldosteronism, it's really hard to talk and write at the same time. We'll talk about aldosteronism as well. It's basically chronic oversecretion of aldosterone due to stress response. If you took a sample of somebody with aldosteronism's urine, you would find extra acidic and potassium-rich urine because those are the two ions that are being traded for sodium. So, aldosterone and other mineralocorticoid release is stimulated by the renin-angiotensin system or by high plasma calcium. So this is also a way that you can, plasma potassium. So this is also a way to scrub extra potassium out of the blood. So if you saw an increase, so let's say somebody's plasma potassium rose, you would expect an increase in aldosterone secretion. And if you saw an increase in aldosterone secretion, you could infer that you would see higher than normal levels of both adrenocorticotropic hormone, so ACTH, and from the hypothalamus, corticotropin releasing hormone, so CRH. So aldosterone's action is inhibited by something called atrial natriuretic peptide. Interestingly, this is a hormone that is secreted by the heart. And natriuresis refers to the urinating out of sodium. So natriuresis is peeing out sodium. So that's the opposite effect. Aldosterone causes retention of sodium in water. Atrial natriuretic peptide causes the secretion of both water and sodium ions. Okay, so this is a good place to stop because the renin-angiotensin system is a very complex system, and it's basically there to respond to changes in blood pressure or blood volume. And those two things are linked because how hard your blood is pushing on your vessel walls is the pressure, and that corresponds to how much blood is in those tubes, which is the volume. So those two ideas are pretty similar. So the renin-angiotensin system causes aldosterone release by the adrenal cortex, and then you get into this big interlocking set of feedback loops, which we will discuss next time. I'm trying to keep these videos a little bit shorter and in bite-sized chunks. So, thank you for your attention, and I will see you in the next video.